Hi everyone, this is the 10th lecture for your chapter Cell. In the last class, we discussed the last two members of the endomembrane system, lysosomes and vacuoles. We studied the structure, function, and their different types. From today, we begin with the organelles which are not considered in this endomembrane system. Okay, so these organelles, they appear and they function independently. We will be talking about mitochondria, the powerhouse of the cell today. Okay. So in Greek, mitos means thread and chondrion means grain. So it looked like a thread-like grain. Okay, that is why it was named as such, mitochondria. It is one of the few organelles which can be seen through your normal light microscope. Okay, so here, of course, all the organelles, they are colorless except for chloroplast and chromoplast. So to observe different organelles, you have to stain it. Here, mitochondria is stained with Janus green dye. Okay, you must have heard of different dyes like safranin, crystal violet, which we have discussed in prokaryotic cell. Okay, gram staining. So here we are staining with Janus green. With this dye, the mitochondria will be able to see. Okay. For example, these are your cheek cells. Have you ever seen your cheek cells or any animal cells under the microscope? If not, this is exactly how it is done. You take a small swab and then you take it inside your mouth and scrape against your cheek. After scraping, you put the scrapings on a glass slide, on a clean glass slide. You spread it evenly and you add some sort of dye. Okay, mostly it is safranin. And then you will be able to see the cell under the microscope. Here we have dyed the mitochondria with Janus green. So can you see these small granules inside the cheek cells? The largest one, the dot you see, is the nucleus, and the small granules you see are mitochondria. This is the diagrammatical representation of what the mitochondria are supposed to look like in light microscope. Okay. They are the major centers of release of energy. Okay, energy in form of ATP, adenosine triphosphate. Okay, so they are the main centers where ATP is synthesized, hence they are called powerhouse of the cell. They are present in eukaryotes which take part in aerobic respiration. Aerobic respiration is a type of respiration where you utilize oxygen and you completely oxidize the organic compound, okay, the substrate. For example, if you are starting out with glucose, the end product of complete oxidation would be carbon dioxide and water, right? So the organisms, the eukaryotes, which carry out aerobic respiration, in these eukaryotes you will be able to find mitochondria. They are absent in prokaryotes and some anaerobic eukaryotes, as well as your RBCs. Okay, again, like I told you in the last class, RBCs you will find as an exception in many of these organelles, okay, which we will study. This diagram given on the bottom is actually an electron micrograph. Okay, so with light microscopy you can see the mitochondria, but they are still dot-like. Okay, to find out their finer structure, okay, their ultra structure, you do require an electron microscope. All the mitochondria present in a cell are collectively called chondriome. This fact is not actually very much required for your NEET, but just in case if sometimes, I mean, you never know where the question may come from. So for your information, the all mitochondria of a cell together are called chondriome. Okay, from the name itself, even if it is asked sometime and you do not remember, from the name you can make it out. Chondriome has to be related to mitochondria. Their number varies depending upon the cellular activity. If the cell is very active, you will find a lot of mitochondria. If the cell does not perform a lot of functions, you will find less number of mitochondria. They are more abundant at the bases of cilia or flagella. The cells which have cilia or flagella, eukaryotes, okay? So they at their base, where the flagella or the cilia is beginning, at those places you will find arrangement of mitochondria, okay? So these mitochondria are present there because movement of cilia and flagella requires ATP, and this ATP has to come quickly. So the mitochondria are present there to provide them with energy. The cells of dormant seeds have very few mitochondria. Just think of the wheat grains that your mother has stored in house, or think of the rice. Okay, so some of these dormant seeds which are not actively growing right now, these seeds have very less or very few mitochondria, and those of germinating seeds have several mitochondria, because the seeds which are germinating have to synthesize many structures, have to divide a lot of times to form nice roots, nice leaves, right, to become a proper sapling. So germinating seeds, they will have several mitochondria present inside their cells. One more thing. Just because the seeds are dormant does not mean they are not respiring. Okay, seeds, though they are dormant, their respiration rate is very, very, very low, but they are very much alive. Okay, the seeds are actually alive. In green plant cells, the number of mitochondria is mostly less compared to animal cells or non-green plant cells. Normally, when you say green plant cell, think about the chlorenchyma or the cells you find in leaves or green colored stem. Okay, but now think of the cells from the root. Okay, cells from root, or maybe even cells from, let us say, flowers and fruits. So these cells will not have chloroplast. Okay, so we can divide the cells into two parts: the green plant cells and the non-green plant cells. The green plant cells have lesser amount of mitochondria because in those cells the chloroplast is present for ATP production. Okay, so mitochondria are required less. Compared to that, in non-green plant cells and animal cells, mitochondria are found in abundance. In absorptive and secretory cells, they lie in peripheral cytoplasm. Okay, so in the cells which are doing the function of absorption, they are absorbing something. Think of your intestinal cell lining. Okay, in your intestine, the cells will be absorbing nutrition, right? So in such kind of absorptive cells, and then secretory cells, the cells which are secreting something, they will have mitochondria lined at their periphery. Let me just draw very quickly. For example, this is the cell. Fine. And this is the nucleus. This is the surface where absorption is taking place or the secretion is taking place. Okay, secretion will happen by exocytosis. Okay, exocytosis, these are the vesicles which are getting fused, and all of that protein, all of that secretion will be secreted outside. Fine. So in such cells, the mitochondria will be found on periphery, on the sides, so as to not hinder the process of absorption or hinder the process of secretion. Okay, so mitochondria will be present on the periphery while the absorption or secretion happens on the upper layer, okay, on the upper membrane. Fine. During nuclear division, more number of mitochondria come to lie around the spindle fibers. Just try to remember, if you remember spindle fibers, okay, these spindle fibers which you see in this diagram are shown in green color. Okay, they are actually labeled with green fluorescent protein. So when you look at them under the fluorescent microscope, they will appear green. Okay, so these green colored fibers that you see are actually microtubules or your spindle fibers. They arise from the centrioles, okay, which are present at the poles. So wherever there are a lot of such microtubule fibers or spindle fibers, mitochondria, which are shown here in red, okay, whatever red you are seeing is mitochondria of the cell. So they come and lie around such spindle fibers. We will see when we discuss cytoskeleton that this is because um, when you have to form a mitotic spindle or you have to break a mitotic spindle, ATP is required. Okay, so this ATP will come from the mitochondria, and these mitochondria are providing the required energy for cell division here. Okay, cell division or division of the nuclear material, which is chromosomes. Okay, the blue colored one you see are the chromosomes aligned at the metaphasic plate. In muscle fibers, they occur in rows between the contractile elements. This is the histological, what you can say, photograph or microscopic photograph of a muscle cell. Okay, and that too a striated muscle. Striated muscle, skeletal muscle, voluntary muscle, everything is one and the same. So this is the actual diagram, okay, actual photograph rather, where you can see these are the muscle fibers, and in between the muscle fibers, the green colored ones you are seeing are your mitochondria. Okay, and whatever the red colored bands you see are actually what you might have learned in locomotion and movement, okay, that chapter. Do you remember that there are A bands, then I band? I band is where the thick filaments are absent, and A band is where thick and thin filaments are present. Then in between you have H zone where thin filaments are absent. So if you try and look here, this is the H zone where only thick filaments are present. Okay, thin filaments are absent around it. Then this is the region where both the type of filaments are present, and then this is the I band where only the thin filaments will be present. Okay, so this kind of structure is of a striated muscle. So in a striated muscle, striated literally means striped because you can see stripes, right? So in striated muscles, you find a lot of mitochondria present between such filaments. A bunch of mitochondria are also found in sperm, especially in the sperm midpiece. Okay, for example, this is a sperm. Okay, its head. The head has this huge purple colored nucleus, and this is the acrosome. Remember, acrosome is made up of Golgi body, right? So this is the Golgi body or acrosome with a lot of enzymes. This here is the nucleus. At the base of nucleus, there is this middle piece which starts. Okay, this middle piece has different filaments, protein in between, what you see here. But surrounding that, the yellow or orange colored structures you are seeing are actually mitochondria. These mitochondria form such kind of rings, okay, C-shaped rings around the neck, and these provide ATP for movement of this tail. Okay, the sperm also has this tail with which it will be able to move. So mitochondria provide ATP for the movement of this tail. Now let us talk about shape, size, and structure of mitochondria. If you say shape, they do not have any specific, only one type of shape. They are normally cylindrical or cigar shaped. They can also be spherical shaped. Okay, so this is the basic shapes. Then length. We talk about the length. They are 1 to 4 micron in size, and diameter is 0.2 to 1 micron. Okay, their chemical composition, what they are made up of biochemically. So mitochondria is made up of proteins about 60 to 70%. Okay, so if you take a mitochondria, you make a chutney and you find out that what are its components, then 60 to 70% of it will be protein, followed by lipids, which are 25 to 35%, followed by RNA, which is 5 to 7%, okay, and has a little quantity of DNA. Then let us talk about the ultrastructure. I told you ultrastructure we were only able to find out after we figured out how to use an electron microscope, okay, and through the electron microscope we were able to see these finer structures, okay. Ultrastructure meaning the detailed structures. So mitochondrion consists of two membranes, outer and inner. Each membrane is 60 to 75 angstrom thick, okay. So here the pink colored membrane you see is the outer membrane, and the yellow or orange colored membrane you see is the inner membrane. So mitochondria, this is the first organelle you are studying which has two membranes, okay. So mitochondria has two membranes, the outer and the inner. Now let us discuss a little bit detail about what these membranes are and how they look like. Fine. The outer membrane is very smooth. Okay, this outermost membrane is smooth. It is permeable to a lot of metabolites. Okay, it is permeable, meaning it will give permission for entry to a lot of molecules. It has protein channels called porins for the same. Okay, so this outer membrane is zoomed in and shown here. So this is the membrane on which you can see these teal colored, okay, or green colored channels. These protein channels are called porins. Okay, just remember pore, P-O-R-E, right? Pore means hole. So these channels are actually holes. They do not close and they do not open. They always stay open. Okay. So these are the porins through which all the hydrophilic molecules like glucose or pyruvate, they cannot come inside the mitochondria through this lipid membrane, because lipid does not allow hydrophilic molecules to move. So through this porin a lot of molecules can come inside. Fine. So because of these porins and other protein channels and transporters, the outer membrane is permissible or permeable to a lot of molecules. It has some enzymes for fatty acid synthesis. It has less protein compared to the inner mitochondrial membrane. Just look at this diagram and remember this fact. Okay, if you see in the outer membrane, you are still able to see the phospholipid bilayer, and in between you are able to see the proteins, right? But when you look at the inner membrane, there are so many proteins and very less amount of membrane you are actually able to see, right? So from this comparison itself, you must be able to realize that the outer membrane has less amount of protein compared to the inner membrane. Now let us talk about the inner membrane. Its protein content is high. How high? Then 70 to 75% of the whole membrane is made up of protein. Can you imagine? So much of protein is present in the inner membrane. It is permeable only to some metabolites. Unlike the outer membrane, inner membrane is very specific. Okay, it will not allow everything to enter. So it is very specific or highly selective about what it will allow to enter the mitochondria. It has infoldings. Okay, these folds that you see, they are called infoldings. These are just the infoldings of inner membrane. Okay, so these inner infoldings are called cristae. Okay, these are all called cristae. What the function of cristae is, it increases the surface area for activity. Fine. So inside the mitochondria, because of these cristae, there will be so much of surface area that it will be able to metabolize things better. Okay, all the reactions can happen properly if there is lot of surface area. One more thing in this diagram, the mitochondria also has its own DNA. This DNA is circular, and because it is a DNA, always remember it is double stranded. Okay, so double stranded DNA, mitochondrial DNA. The density of cristae, now we are just talking about the inner membrane. The number, okay, and the density of cristae, it indicates how the intensity of activity done by the cell. Okay, how much active the cell is can be found out first thing from the number of mitochondria, and second thing from finding out how many number of cristae are present in each mitochondria. Fine. Enzymes of electron transport chain are located in the inner membrane. Okay, so if this is the inner membrane which has these enfoldings, the enzymes of electron transport chain are located on the inner membrane. The cristae, which are these, they possess small lollipop-like particles called elementary particles. Okay, this is the electron micrograph. You must be able to see these small dots, right? So these small dots, and they have a small stalk also, right? So this is the diagrammatic structure of the same, and then the zoomed in version. Okay, what are these particles exactly? So these are called elementary particles, or were called elementary particles. We normally use the term FOF1 particles or FOF1 ATPase. Okay, they are also called oxysomes. The only reason I'm mentioning these names is sometime if in any kind of test you are asked, then you will be able to answer the question. Okay, but if you know the name FOF1 particle or FOF1 ATPase, that is enough. The FOF1 particles have a head. So this here, the blue colored one is the head. Then it has a stalk and it also has a base. Okay. How this functions is the protons. Okay, we will be discussing this in respiration more detail. Okay, but for now, just know that the base is actually a proton channel. Okay, so the protons, H plus ions, they move through this base and through the stalk, and they go towards the place or the side which has lower concentration of protons. Fine. So protons are moving from their higher concentration to lower concentration. They move through these proton channels, which are the base of uh, FOF1 particle. Okay, then you have the stalk, which is connecting it to this head. Okay, this head actually acts as an ATPase. Okay, as the protons move, this base will rotate like a merry-go-round. As the base rotates, the head also rotates, and when the head rotates, it will be making from ADP into ATP. Okay, so ADP plus inorganic phosphate will give you ATP because of this movement. They function as ATPases, which are center for ATP synthesis during oxidative phosphorylation. You will also find these kind of structures, FOF1 particles, in chloroplast as well. Okay, so we will discuss that in detail when we talk about photosynthesis. Okay, and this is the comparison between the outer membrane and the inner membrane. The outer membrane is smooth, while the inner membrane has a lot of proteins. Okay, so it will appear a little bit uneven. The outer membrane has porine channels. Okay, this porins, they allow a lot of things to pass inside. Okay, so it is permeable to most biochemicals. The inner membrane has carriers and other transport proteins, but it is selectively permeable. The enzymes in the outer membrane are fewer, while the inner membrane contains a lot of enzymes. The foldings are absent in outer membrane, while the inner mitochondrial membrane has large number of enfoldings called cristae. Cholesterol and other lipids are present. Cardiolipins are absent. Okay, cardiolipins are a type of phospholipids. Okay, they are double phospholipids even. We shall discuss if required when we talk about biomolecules. Okay, for now, if you want to remember, well and good. If not, it is fine. Okay, so cardiolipins are absent, while in the inner layer, cardiolipins are present. And the last part is electron transport chain or electron transport system is absent on the outer membrane while it is present in the inner membrane. If you have any more differences, please feel free to add to it. Okay. Right. Now there are many metabolic pathways which occur in the mitochondria. The very famous one is the TCA cycle, right? So TCA cycle, it starts with the pyruvate coming inside the mitochondria. This pyruvate is converted into what we call acetyl-CoA, right? So this acetyl-CoA, it will get joined with the oxaloacetate. So acetyl-CoA plus oxaloacetate will give you citric acid, isocitric acid, alpha-ketoglutarate, and so on and so forth. So this here is the tricarboxylic acid cycle, right? So its major metabolic pathway would be this tricarboxylic acid cycle or Krebs cycle. Fine. From this Krebs cycle, you will be able to see that this alpha ketoglutarate, okay, it can even go to making glutamate. So it is used for synthesis of some other amino acids as well. Then succinyl CoA will be used or can be used to make porphyrins like heme. Okay, heme group, hemoglobin has heme, then chlorophyll has heme, right? So all of these heme groups are actually synthesized because of the intermediate you get from mitochondria, which is succinyl CoA. Then different other formation, okay, synthesis also happens inside the mitochondria. Like here you can see the tetrahydrofolate, okay, and this tetrahydrofolate, which is actually folic acid, okay. Folic acid is vitamin B9. So it will be used to make glutathione, purines, thymidine, okay, different kinds of nucleotides, different kinds of molecules. So it is not necessary all of these will be synthesized in the mitochondria, but the mitochondria has a lot of precursors, okay, or intermediates which are used for other synthetic pathways. So TCA cycle or Krebs cycle, oxidation of fatty acid. Remember this term, beta oxidation of fatty acids. Okay, remember all those fatty acids you saw on the phosphatidyl choline and phosphatidyl inositol tails. Okay, in phospholipids, you draw two tails, right? So those tails are actually fatty acids. These fatty acids get oxidized in mitochondria. Okay. Then electron transport chain occurs in mitochondria, followed by oxidative phosphorylation, which is production of ATP, transamination, which I just explained, from alpha ketoglutarate, which is a keto acid. When you add an amino group, it will get converted into glutamate, which is an amino acid, right? So such kind of transamination, synthesis of urea and intermediates for other pathways occurs in mitochondria. So it is very, very, very important for metabolism. Now we were talking about the outer and the inner membrane. Between these membranes, there is some space. Okay, so this you can see is the outer membrane, and this here is the inner membrane. The space between the two is called intermembrane space or perimitochondrial space. Okay, so here whatever this light colored portion you are seeing is the intermembrane space, okay, or perimitochondrial space. It is about 60 to 100 angstrom wide, okay, and it extends into spaces of cristae. You can see that this intermembrane space is present even in between such cristae. Okay, it contains very few enzymes. It has a few enzymes, okay, not many, but that space is also important. We will see this when we talk about respiration. Okay, the proton concentration will increase in this intermembrane space, and from here they will be transported towards the mitochondrial matrix, and during that transport ATP will be synthesized. So this intermembrane space is very, very important. Now that was all about the intermembrane space, which is the space between the two membranes. What about the inner space, okay, which is covered by the inner membrane? So this space is actually called matrix. It is a fluid, okay, just like your cytoplasm. It has proteins, it has ribosomes, which are of 70S type. Okay, it has ribosomes, which are 70S type. It has RNA, okay, it will be mRNA, tRNA, rRNA. It has mitochondrial DNA present inside the matrix. It has enzymes for the Krebs cycle, okay, the citric acid cycle enzymes are present in the matrix. Then amino acid synthesis and fatty acid metabolism will all occur in the matrix. It also has a few crystals of calcium phosphate and manganese. Okay. Now let us talk about autonomy of mitochondria. Now these organelles, they do not come from any other organelles. Okay, like how we discussed that Golgi body comes from maybe endoplasmic reticulum, right? Then smooth endoplasmic reticulum comes from rough endoplasmic reticulum, which has left all of its ribosomes, right? Then vesicles or lysosomes, they come from Golgi body. Vacuoles also come from Golgi body or ER. Okay, so we have studied that one or the other organelle comes from some other organelle. Okay, but here in case of mitochondria, these mitochondria, they replicate and they exist independently. They have their own DNA which can replicate independently. Okay, so their DNA can also get replicated on their own. Then this mDNA, which means mitochondrial DNA, produces its own mRNA, tRNA and rRNA. Okay, messenger RNA, transfer RNA, ribosomal RNA. They have their own ribosomes, which are of 70S type. Okay, so this rRNA will be present in this 70S ribosomes. Okay, then they will read the mRNA, which is also called transcript, and with help of tRNA they will bring the different amino acids according to the mRNA and start producing new proteins. So these ribosomes, they are used to synthesize some, okay, focus on the word some, structural proteins and enzymes of the mitochondria. Not all, only some. However, most of the mitochondrial proteins are encoded in the nuclear DNA. New mitochondria develop by binary fission or fusion of pre-existing mitochondria. Okay, if you have heard of this term binary fission, okay, in bacteria and other simpler organisms, you find binary fission as a mode of asexual reproduction. Here also, in case of mitochondria, you see binary fission, and with this binary fission you get new mitochondria. Okay, so mitochondria come from pre-existing mitochondria. However, they are not fully autonomous. Autonomous means self-reliant, independent. They are not fully independent. Both their structure and functioning are partially controlled by the nucleus. Okay, their structure and their functioning is somewhat controlled by the nucleus, and availability of material from the cytoplasm, whether the materials for its functioning are available or not, it depends on the cytoplasm. So let us see here mitochondrial fission. Okay, fission is when this is an original mitochondria. You will find a contractile ring forming and then it will divide into two. So now you have two mitochondria. This is called fission. Okay. There is also fusion in times of stress, when there is stress or when you have to work way harder. In such cases, two mitochondria can come together and they can actually fuse to make one big mitochondria. Okay, this is called mitochondrial fusion. Now, there is a theory called endosymbiotic theory. You know symbiosis. Symbiosis is friendship, right? Both the parties involved in this friendship will be benefited. So here you have endosymbiotic theory. What does this theory say? It says that the oldest eukaryotes did not have mitochondria. Okay, they just had this nucleus and they had the this endoplasmic reticulum, right? Then what happened? One day there was some bacteria, okay, which was aerobic bacterium. Some say it was purple bacteria, some say it was aerobic bacteria. In the end, it was the one which was using oxygen and doing aerobic respiration. Okay, so this bacteria, it entered in the cell. Okay, and as it entered in the cell, it found that the cell will provide it with nutrition and it can help the cell by providing it with ATP. So they found this symbiotic relationship, and later on this aerobic bacterium inside the cell, after a long time, it became mitochondria. Okay, same is the case with chloroplast. Chloroplast must have been a photosynthetic bacterium which entered the cell and became symbiotic with it. Okay, it decided to produce food. Okay, for it, and in turn, it got protection from the cell. Okay, so this is the endosymbiotic theory, which shows that the mitochondria might have been a separate bacteria in the beginning, but they entered the cell at some point of time, and now they have become a integral part of our cells. Okay, also mitochondria entered first because even plant and animal, both cells have mitochondria, and the chloroplast must have entered later on. Okay, because animal cells do not have chloroplast, only plant cells do. Fine. So this is the endosymbiotic theory, which proves that why this mitochondria is semi-autonomous. Semi means half. Autonomous means self-reliant. Okay, it is semi-autonomous. It can function on its own, but it needs some help from the nucleus. Now we come to the last part, which are the functions of mitochondria. This is just a quick recap. Okay, so we'll talk about the first and the foremost, which is complete oxidation of respiratory substrate. Okay, so it is important for aerobic respiration of respiratory substrate. Respiratory substrate meaning the molecules which can be oxidized to get ATP. It includes glucose, fatty acid, glycerol, etc. Okay, so these are all respiratory substrates. It completely oxidizes them to carbon dioxide and water inside the mitochondria. The energy liberated by aerobic respiration is stored in form of reduced coenzymes like NADH and FADH2. These coenzymes are finally used to make ATP, okay, as we shall see in respiration. ATP helps in energy requiring processes like muscle contraction. Okay, if you want to contract your muscle, you require a lot of ATP. So in muscle contraction, nerve impulse conduction, okay, remember action potentials. So even action potentials, they have sodium potassium ATPases, which use ATP to pump sodium and potassium, right? So nerve impulse conduction also requires ATP. Then biosynthesis of new molecules requires ATP. Membrane transport requires ATP. Just think about active transport, then endocytosis, exocytosis, receptor mediated endocytosis, all of these things require ATP. Then cell division requires ATP. Okay, so all of these energy requiring processes are occurring, okay, or they are helped by mitochondria because of mitochondria. Mitochondria provides important intermediates as we saw in that metabolism slide. Okay, so it provides important intermediates. Intermediates matlab beach wale molecules. Okay, some molecules which are present in the whole pathway, and this molecules will be used or can be used rather to synthesize new biomolecules like chlorophyll and cytochromes, which I told you they are synthesized from heme, okay, the porphyrin. So chlorophyll, cytochromes, then pyrimidines, alkaloids, and a lot of other things. Synthesis of many amino acid occurs in the mitochondria. At first, the two amino acids are made, okay. The first amino acid to be made is glutamic acid. It is synthesized from alpha ketoglutarate acid. Alpha Kg you must have heard in TCA cycle, right? Then you have aspartic acid. Aspartic acid is synthesized from oxaloacetic acid, which again you must have heard in TCA cycle, right? From this glutamic acid and aspartic acid, now these amino acids will be used to make newer amino acids. How? So different keto acids like oxaloacetate or alpha ketoglutarate, to them the amino group will be transferred and new amino acid will form. Okay, so by transamination, okay, an organism generally receives mitochondria from its mother. So this is called maternal inheritance. Think of your mother, then of her mother, then her mother. Okay, think of the mother of all human beings. Fine. So we still share mitochondria from her, okay, from the common mother. Why? So when you look at the egg cell, okay, this is an egg cell or an ovum. The egg cell has a lot of mitochondria. Even the sperm has mitochondria in its middle part. But after fusion, after fertilization, only the head or the nucleus of sperm can enter inside the cell. So the sperm or the male, okay, the father, he does not contribute in giving the mitochondria. Fine. Only the egg contributes mitochondria to the zygote. So this mitochondria will have DNA which was same with the mother's mitochondrial DNA. So this is the reason why it is called maternal inheritance. This mitochondrial DNA you can only get from your mother and never from your father. You might study or you might get a question like this. This is called pedigree analysis, which you might already have studied, okay, in genetics. So let me just quickly explain how it is, okay. So sometime if you have to solve such kind of pedigree analysis, your brain would work on in this direction specifically, okay. So here you have parents. The round everywhere where you see the circle, okay, that circle is the lady, the woman, and square is a man. The filled circle means the lady who has some trait, okay, some defect in mitochondria, let us say. So that defect in mitochondrial DNA is shown in with filled colors, okay. In case of ladies, it is shown with pink. In case of men, it is shown with blue. So the parents, the father does not have this specific mitochondrial defect in the DNA, okay, but the mother has some mitochondrial DNA defect. Their children will all get the mitochondria from their mother. So the three girls, 1, 2, and 3, all three of them have their mother's DNA, mitochondrial DNA, and their son also has the mitochondrial DNA. So um, all the four children of this couple has the mitochondrial DNA of mother, which is defective. The three sisters get married to a normal man, okay, and you will find that even their children have such mitochondrial DNA defect. Okay, you can see this and this, right? Now their children, the F2 generation, they also get married, and let us see their children now. Okay, so if it is a lady, a girl who has this mitochondrial DNA defect, it will be passed on to her children. If it is a man who has this mitochondrial DNA defect, this man does not contribute to his children's mitochondria. Okay, so his mitochondrial DNA will never be passed on to the children because he does not contribute in the mitochondria. Okay, to give mitochondria to the embryo. Same thing here, if the lady has this mitochondrial DNA defect, the children, all of the children will have it. But if the man has this mitochondrial DNA defect and the woman is normal, okay, without this genetic trait or without this defect, the children will not have the defect. Fine. So this is what is meant by maternal inheritance, and here we end today's class. Okay. If you have any doubts, let me know and see you next time.